So today I will discuss about renal physiology. We need to know that what things they are absorbed from proximal convoluted tubule. So it is to tell you that everything is reabsorbed from proximal convoluted tubule, including glucose, amino acids, phosphates, bicarb, water, sodium, and potassium. Then further down in the descending limb of loop of Henle, sodium chloride, potassium, and water is reabsorbed. Then if you go further down, then magnesium and calcium is also reabsorbed. And if you see the tip of the loop of Henle, here the fluid is maximally concentrated because water is being reabsorbed. Then as we go up again in the ascending limb of loop of Henle, we can see here that water, it is not reabsorbed at all. It remains within the tubule, while the solutes or the electrolytes, they are reabsorbed, like magnesium, calcium, they go outside. So this is an important question that come in the exam, that which part of the glomerulus or nephron is impermeable to water. So it is basically ascending limb of loop of Henle. Now, these are the main processes which are involved in the formation of urine. This is glomerular filtration, tubular reabsorption, and tubular secretion. In filtration, water and solutes smaller than proteins, they are forced through the capillary walls and they go into glomerulus, and through glomerulus into renal tubule. Then after that, then some wanted substances, they are reabsorbed back into blood, like water, glucose, amino acids, and some needed ions. Then there's a process of tubular secretion, like hydrogen, potassium, creatinine, and drugs. They are removed from peritubular blood and again secreted into the nephron. So this is basically that the diuretics, where do they act on nephron? This is very important from exam point of view. Carbonic anhydrase inhibitor, they act on proximal convoluted tubule. Then osmotic and loop diuretics, they act on loop of Henle. Thiazide diuretics, they act on proximal part of distal convoluted tubules. And potassium sparing diuretics like spironolactone, they act on distal part of distal convoluted tubules. ADH, it acts on collecting duct. So this is all about kidney, basic clinical sciences. So after that, some basic things about cardiac physiology. This is action potential. In phase 0, there is rapid depolarization is because of sodium influx. Then early repolarization of phase 1, which is because of potassium efflux. And then phase 2, which is plateau phase, which is because of calcium influx. And phase 3, again because of potassium efflux. So it is easy to remember that phase 1 of repolarization and phase 3 of repolarization, both are because of potassium efflux. And then final stage is the restoration of resting membrane potential. Then few definitions, they are important from examination point of view. Ejection fraction is actually the ratio between stroke volume and end diastolic volume. Then stroke volume is actually the difference between end diastolic volume and end systolic volume. And cardiac output is equal to heart rate into stroke volume. Then another important topic is atherosclerosis. Usually it is a very complex process, but as far as the exam of mrcP 1 is concerned, I will discuss just 3 steps. In the first step, there is injury to the blood vessel because of turbulent flow of blood. As a result of this, the monocytes, they get differentiated into macrophages. Then in the second step, low density lipoprotein, it moves towards these macrophages, and these macrophages, they engulf this low density lipoprotein and they become foam cells. And in the last step, there is proliferation of smooth muscles, and then they move to intima to form the atherosclerotic plaque. Then after that, there is another important exam question, that is about actin and myosin filament. We know that actin is a thin filament and myosin is the thick filament, and by interaction of these two filaments, cardiac muscles or skeletal muscles, they contract. Now, actin or thin filament, it is wrapped by a strand of tropomyosin. Function of tropomyosin wrapping is to control the contraction and relaxation of skeletal or cardiac muscles. Now, this tropomyosin has got 3 other proteins which are called troponin T, troponin I, and troponin C. Now, troponin T, it actually is attached to tropomyosin, and troponin I, it inhibits actin filament to interact with myosin filament, and thus it inhibits the contraction of muscle when it is in the relaxed phase. So it prevents the unwanted joining of actin filament with myosin filament. Then after that, troponin C, which binds with calcium. So troponin T binds with tropomyosin. Troponin I, it actually inhibits the combination or fusion of actin and myosin filament. And troponin C, it binds to calcium. And all these three troponins, they are attached to tropomyosin, and tropomyosin is wrapped around thin actin filaments. And function of tropomyosin and these three troponin is to control the contraction and relaxation phase of muscles.